निरी वेबिनार हामीले आजभन्दा 3 वर्ष पहिला सुरु गरेका थियौँ। अहिले हामी चौथो वर्षमा छौँ। हामीले नियमित रूपमा विश्वभरिका नेपालीहरूबाट अथवा अन्य वैज्ञानिक तथा अनुसन्धानकर्ताहरूबाट गरिएको अनुसन्धानलाई आम मानिसहरू बिच पुर्याउने हेतुले निरी वेबिनार सुरु गरेका हौँ। आज हामीले यमन थापालाई निम्ताएका छौँ। यमन थापा काठमाडौँको सेन्ट जेभियर कलेजबाट +2 गरेर 2018मा अन्डर ग्रेजुएट डिग्रीको लागि अमेरिका आउनुभएको हो। न्यू जर्सीमा अवस्थित अमेरिकाको न्यू जर्सीमा अवस्थित कार्लवेट युनिभर्सिटीबाट उहाँले अन्डर ग्रेजुएट गर्नुभयो, स्नातक गर्नुभयो बायोलोजीमा। र त्यसपछि उहाँ चाहिँ विश्वविख्यात ब्रोड इन्स्टिच्युट भन्ने छ, जुन एमआइटी र हार्वर्डको सहकार्यमा निर्माण गरेको इन्स्टिच्युट हो। त्यहाँबाट उहाँले समर इन्टर्नसिप गर्नुभयो। त्यसपछि उहाँले अहिले कोल्ड स्प्रिङ हार्बर ल्याबोरेटरी भन्ने लङ आइल्यान्ड न्युयोर्कमा अवस्थित विश्व प्रसिद्ध इन्स्टिच्युटमा पिएचडी गर्दै हुनुहुन्छ, विद्यावार्ती गर्दै हुनुहुन्छ। अत्यन्त चाखलाग्दो कुरा के छ भने हामी जस्तै मुसाहरूले पनि गीत गाउने गर्छन्, एकअर्कालाई रेस्पोन्ड गर्ने गर्छन्। त्यसले आजको वेबिनारमा हामीले गीत गाउने मुसाहरूको बारेमा सुन्नेछौँ उहाँबाट, यमन थापाबाट। मैले छुटाउनै नहुने एउटा कुरा के छ भने आजभन्दा 2 महिना अगाडि म कोल्ड स्प्रिङ हार्बर ल्याबोरेटरीमा एउटा तालिमको लागि 2 हप्ते तालिम अथवा गोष्ठीको लागि गएको थिएँ। त्यहाँ मैले यमन थापालाई भेट्ने मौका पाएँ। उहाँको र मेरो चिन्जान 2 महिना मात्रै लामो छ, तर उहाँलाई भेट्दाखेरि उहाँ नेपाली हुनुहुन्छ, को हुनुहुन्छ होला यो इन्स्टिट्युसनमा भनेर भेट्दाखेरि खोज्दा उहाँलाई भेटेँ। र त्यो नेपालप्रतिको लगाव, यहाँको सिपहरू नेपालमा लैजाने, नेपाललाई कसरी हेल्प गर्न सकिन्छ, नेपालको युथहरूसँग कसरी जोडिन सकिन्छ भन्ने उहाँको त्यो लगाव, उहाँको त्यो कमिटमेन्ट देखेर म एकदमै प्रेरित भएँ। त्यसैले मलाई लाग्छ उहाँलाई कुरा गर्दाखेरि त्यो उहाँले चाहिँ अहिलेको त्यो युथहरूको लागि रिप्रेजेन्ट गर्नुहुन्छ भन्ने मलाई लाग्छ। थ्याङ्क यू सो मच फर इन्सपायरिङ यमन, and thank you so much for agreeing to give a presentations in NIRI webinar. Over to you. 30-40 मिनटभित्रमा हजुरले प्रेजेन्टेसन सक्नुभयो भने हामी क्वेसन एन्सरतिर जानेछौँ। थ्याङ्क यू सो मच एन्ड इट्स ओभर टु यु नाउ।
थ्याङ्क यू सो मच। मलाई पनि एकदमै खुसी लाग्यो हजुरलाई भेट्दा। अनि यो निरी इनिसिएटिभले सकेसम्म बायो इन्फर्मेटिक्सको रूपले भए पनि नेपालीहरूलाई रिसर्चसँग इन्ट्रोड्युस गरेर जुन साइन्स एडभान्स गरिरहेछ त्यो एकदम खुसी लाग्यो। हजुरलाई एकदमै थ्याङ्क यू मलाई इन्भाइट गर्नुभएकोमा। सो आज चाहिँ म पिएचडीमा म 3rd इयर हो अनि मैले रिसर्च चाहिँ 1.5 इयर गरिसकेँ। मैले त्यही 1 वर्ष आदिमा कति रिसर्च गरेँ, के गरिरहेको छु भनेर सेयर गर्छु आज चाहिँ। म चाहिँ 2 जनाको ल्याबमा छु। मेरो एउटा एडभाइजरको नाम चाहिँ डाक्टर अर्करूप ब्यानर्जी, अर्को एडभाइजर चाहिँ डाक्टर बेन्जामिन काउली। एकजना चाहिँ एक्सपेरिमेन्टलिस्ट हुनुहुन्छ प्राइमेरीली, अर्को चाहिँ कम्पुटेसनल हुनुहुन्छ। सो मैले चाहिँ एक्सपेरिमेन्ट अनि डेटा एनालिसिस, मेसिन लर्निङ युज गरेर एउटा एकदमै राम्रो बिहेभियर, इनेट बिहेभियर स्टडी गरिरहेको छु। ओके, स्टार्ट गरेँ त। ओके, सो हामी एभ्रिडे लाइफमा वी इन्टरेक्ट विथ एनिमल्स होइन, अनि एनिमल्सहरूको एकदमै फ्लेक्सिबल बिहेभियर हुन्छ होइन। उनीहरूको बडीमा सेल्स, अर्गन्स सबै हुन्छ अनि दे रेगुलेट देयर बिहेभियर, दे हेभ एन इन्टर्नल स्टेट। त्यसमाथि पनि अर्को एनिमल आयो भने अथवा इन्भाइरोमेन्टमा केही भयो भने दिज एनिमल्स आर एबल टु एड्याप्ट देयर बिहेभियर अकर्डिङ टु द इन्भाइरोमेन्ट। अनि यो कुराले चाहिँ उनीहरूको सर्भाइभलमा एकदमै हेल्प गर्छ। फर इक्जाम्पल मुसाहरू लाइक अस ह्युमन्स, दे हेभ अ स्लिप वेक साइकल होइन। वेक साइकलमा चाहिँ दे आर भेरी एक्टिभ, दे मुभ अराउन्ड, दे गो अराउन्ड। when they're sleeping they're inactive, right? यो बिहेभियर चाहिँ इन्टर्नली रेगुलेट हुन्छ। सो हाम्रो सेल्समा there are these feedback loops, these cellular biochemical reactions that take 24 hours. And this regulates their behavior. But like यो बिहेभियर इन्टरनली रेगुलेट भए पनि एक्सटर्नल स्टिमुलाई इन्भाइरोमेन्टमा केही भयो भने हामीले यो बाइपास गर्न सक्छौँ, राइट? सो हामीलाई कहिलेकाहीँ निन्द्रा लागिरहेको हुन्छ, तर बाघ आयो भने हामीलाई निन्द्रा लाग्दैन, होइन? आवर बडी इज गोइङ टु try to run away from the tiger. So this is a flexible behavior. Another really cool thing about this behavior is हाम्रो यो behaviorलाई हामीले several like time scales मा study गर्न सक्छौँ. For example, we can look at a behavior. हामीले जब like if we touch something that's really hot, तातो कुरा समातेपछि हामीलाई झट्ट we're going to remove our hands. So त्यो चाहिँ milliseconds मा हुन्छ. Similarly हाम्रो यो सुत्ने उठ्ने काम गर्ने सुत्ने उठ्ने त्यो साइकल चाहिँ डे स्केलमा हुन्छ. So in this way behavior spans from days to milliseconds. अनि the cool thing is like day scaleमा चाहिँ नि it's very heavily studied. So हाम्रो journals पनि छ that is dedicated to circadian rhythm. Circadian मतलब यो 12-hour active inactive rhythms. On the other hand, हाम्रो visual processing हामीले कसरी देख्छ, हामीले कसरी त्यो process गर्छ, हैन? Things like that are in the opposite end of the scale that happens within seconds and milliseconds. तर यो बिचको time scale हुन्छ नि, त्यो minute को time scale and hours को time scale, those time scales are not that well studied. So मेरो PhD ले चाहिँ यो time scale मा focus गर्छ. Okay. And to study this, we have a really cool tool. So हाम्रो एउटा राजेन सरले अलिकति भन्नुभएको थियो पहिला नै, मुसाहरू, यो मुसाहरू चाहिँ कोस्टारिकाबाट आउने हो. हामीले दैनिक रूपमा इन्टरेक्सन गर्ने मुसाहरू जुन हामीले देखेको छौँ, त्यो मुसा जस्तै दे आर भेरी क्लोजली रिलेटेड एभोल्युसनरी. तर यिनीहरूले चाहिँ नि गाउन सक्छन्. सो उनीहरूले चाहिँ नि they sound like insects. अनि they can sing, which is a really cool tool because यो उनीहरूको त्यो vocalization, उनीहरूको त्यो speech study गरेर we can look at this adaptive behavior at several time scales. So I'll tell you more about this mice and their really cool behavior. Number one is your behavior चाहिँ innate हो. उनीहरूलाई गाउन सिकाउनु पर्दैन, उनीहरू आफै गाउँछन्. अनि usually के हुन्छ भन्दा चाहिँ when we study a mice, we like lab mouse, we train them and we reward them, right? उनीहरूलाई हामीले एउटा बटन प्रेस गर्न सिकाउँछौँ अथवा लिभर प्रेस गर्न सिकाउँछौँ, अनि उनीहरूले त्यो गरिसकेपछि हामीले उनीहरूलाई खाना दिन्छौँ, हैन? रिवार्ड दिन्छौँ. हामीले कुकुरहरू ट्रेन गर्दा नि त्यस्तै गर्छौँ, हैन? रिइन्फोर्स गर्ने कुनै बिहेभियर. So in the same way, the research related to aggression, motivation all require a cue and reward. तर हाम्रो यो singing mice को केसमा चाहिँ नि we don't have to reward them, they just sing, they just know how to sing and they just sing. Here यो एउटा example है, like चाहिँ spectrogram भन्छ. यो चाहिँ 8 सेकेन्डको spectrogram हो. यो जुन त्यो लाइनहरू छ येलोमा, those are notes. उनीहरूले notes बनाउने हो. अनि यो चाहिँ y-axis मा frequency हो. So what you can see is like first मा उनीहरू सानो 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 notes बनाउँछन्, अनि त्यसपछि as the song progresses the notes get longer. The other really cool thing is यो एकदमै cool छ. उनीहरू एक्लै हुँदा isolation मा पनि गाउँछन्, तर उनीहरूले एक अर्कासँग पनि interact गर्न सक्छन्. So when one mouse sings, the other one replies within 0.5 seconds, as quickly as human interaction. So this is a really cool feature. The other thing is that your behavior la हामीले multiple time scales मा study गर्न मिल्यो. For example, उनीहरूको notes milliseconds मा हुन्छ, उनीहरूको songs seconds मा हुन्छ, 7 seconds long हुन्छ. त्यसपछि हामीले यसलाई minute मा हेर्न मिल्यो, hours मा हेर्न मिल्यो, अनि days मा हेर्न मिल्यो. In this way we can look at how the biology of an organism can affect its behavior at multiple time scales. Okay, एउटा example छ मुसाहरूले गाएको. I don't know if it's going to play in everyone's end, but I'm just going to play it. So उनीहरूले यसरी, they sound like insects. एउटाले गायो and you can see त्यो एउटा posture लिएर गाउँछ, अनि एउटाको end भइसकेपछि अर्कोले reply गर्छ, हैन? You can see their mouth moving. and they're going back and forth. So you would say हाम्रो human interaction जस्तै back and forth भयो हैन. Okay, this is an example. हामी मान्छेहरू बोल्दा पनि we do back and forth, right? अनि environment को बेसिसमा हाम्रो त्यो behavior change हुन्छ. So singing mice is a great model to study human interaction. अनि हाम्रो धेरै रोगहरू Parkinson's अरू मोटर डेफिसिट भएको रोगहरूमा हाम्रो स्पीच इम्पेयरमेन्ट हुन्छ, हाम्रो मोटर इम्पेयरमेन्ट हुन्छ. अनि अन्डरस्ट्यान्डिङ द सर्किट अफ दिस बिहेभियर इन माइस हामीले मुसाहरूमा एकदमै मजाले हाम्रो टुल्स एक्सपेरिमेन्टल टुल्स एकदमै राम्ररी अभाइलेबल छ. त्यो पढेर हामीले ह्युमन इन्टरेक्सन पनि बुझ्न सक्छौँ. अनि यो धेरै रोगहरूको ट्रिटमेन्टको रिसर्चमा पनि कन्ट्रिब्युट गर्न सक्छौँ. सो द्याट्स द मोटिभेसन बिहाइन्ड द रिसर्च. सो विथ द्याट मेरो चाहिँ 3टा एम्स छ to go through throughout my PhD. I'm going to discuss some of it with you. So number 1 चाहिँ यो temporal patterning, यो singing, यो behavior छ ल they sing हैन, मुसाहरूले गाउँछन्, तर कसरी गाउँछन्, कतिचोटि गाउँछन् हैन, त्यो अनि त्यो टाइमिङको प्याटर्निङ बुझ्ने चाहिँ मेरो फर्स्ट एम हो. सेकेन्ड एम चाहिँ युजिङ मसिन लर्निङ मोडल मैले चाहिँ म्याथमेटिकली यो बिहेभियरलाई डिस्क्राइब गर्न खोजिरहेको छु. अनि थर्ड चाहिँ रियली द मोस्ट एक्साइटिङ थिङ इज लाइक हाम्रो ब्रेनले लाइक मुसाको ब्रेनले कसरी यो गर्छ हैन, लाइक उसको आफ्नै इन्टर्नल स्टेट छ, तर जब अर्को मुसा आएपछि उसले कसरी त्यो इन्फर्मेसनलाई बायोलोजिकली इन्टिग्रेट गरेर हाउ डज इट चेन्ज इट्स बिहेभियर. सो द्याट्स अनदर रियली एक्साइटिङ थिङ द्याट आइ एम गोइङ टु बी फोकसिङ अन. Okay, for this talk, I'm only going to tell you about M1 and M2. I'm still working in M3. Okay, so my experimental design ko barema bhanchu. So the design and with this, my lab mates are Mike, Martin, and Cliff. They helped me design this experiment. So हाम्रो यो मुसाहरू छ, we put them in an isolated box. अनि उनीहरूले एकअर्कालाई सुन्न सक्दैनन्. So for like days, I acclimate them, and after that, for 5 days, उनीहरूलाई आइसोलेटेड छ. अनि म चाहिँ के गर्न मिल्छ भन्दा चाहिँ उनीहरूको यो इन्टर्नल स्टेटले मात्र उनीहरूले जुन गाउँछन् त्यो स्टडी गर्न मिल्यो. त्यसपछि 5th डेमा मैले त्यो बक्सको डोर खोलिदिन्छु, अनि त्यो खोलेपछि दे क्यान हियर इच अदर, राइट. सो 5 दिनसम्म एक्लै थिए. त्यसपछि ढोका खोलेपछि चाहिँ उनीहरूले एकअर्कालाई सुन्न मिल्यो. त्यसपछि आई पुट देम ब्याक इन द सोलो टु सी हाउ देयर internal rhythm has changed because of social interaction. Okay, so here are the results है त. So मैले चाहिँ यो एउटा रोमा एउटा डेको प्लट हो. यो यहाँदेखि यहाँसम्म लाइट अन हो. सो यिनीहरूले चाहिँ हामी मान्छेहरू जस्तै लाइट बाहिर लाइट हुँदा they're active, right? So you can see what I'm plotting here is songs. So if it's very high here, that means a lot of songs. If it's empty, it means no songs, right? So उनीहरू चाहिँ light on भएपछि they sing a lot, right? So यो चाहिँ हामीलाई diurnal भन्छ. यो चाहिँ circadian rhythm हो हैन. You can appreciate also across mice, they have a rhythm. अनि they are all active during the daytime and they're not active at night, right? Cool. त्यसपछि we can we can zoom in and look into it at a finer resolution. And here you can see, you say hours, this is roughly 12 hours, 13 hours. You can see they're very active in the morning. Like this bout is like 4 hours. And this is at a finer resolution. अनि हामीले चाहिँ एउटा Fourier transform भन्ने टेक्निक छ, त्यो गरेर हेर्यो भने चाहिँ we can see these frequencies at a lower time scale than 12 hours. So we call this ultradian rhythm. Ultradian matlab of a higher frequency than 12. And again, we can look into it even at a finer resolution. We can zoom in further. And again, this is one row is an hour, and each tick is a song, and you can appreciate that they're singing a lot, and each mouse has their own rhythm and so on. Okay, now further zoom in garda, we can look at one song. Like how long is a song? त्यो songमा के information exchange bhairach and so on. Again, finer resolution, look at it at a minute scale. In this way, it spans 7 orders of magnitude. Okay, So naturally, I think one of the things हजुरहरूले सोचिरहनु भयो होला लाइक उनीहरू कसरी गाउँदो रहेछ नि त होइन दिनभरि कतिचोटि गाउँछन् what information are they exchanging? So that's what I asked, right? Like given that they sing throughout the day, how are their songs patterned at a minute scale? So are they just singing randomly? Is there some kind of pattern, right? So here I'm showing you, I'm plotting the mean and the standard deviation. So a pure clock, every 10 minutes tick गर्ने clock भएको भए, we would have zero standard deviation. So it would look like this. It would be a straight line. If it was a random, just random points, then it would follow an exponential distribution, we say. Ani random भयो भने चाहिँ the lines should look like this. So where does our data lie? We looked at our data, we zoomed in at a minute scale, and we saw that उनीहरू एकदमै regularly गाउँदो रहेछन् क्या. Like यो चाहिँ dash line is perfectly 9 minutes, and the colored lines are songs. And what you can appreciate is how time locked it is to 9 minutes. होइन यसको मतलब चाहिँ के हो भन्दा उनीहरू every 9 minutes or every X minutes, एकदमै regularly, एकदम थोरै error, थोरै noise भएर गाउँछन्. And so when we look at the plot, we see that our data lies somewhere in the middle. So it's like an imperfect clock. It's a noisy clock. And if we look at it in all the mice, we see that they have different clock rates, right? So in the morning, एकदमै धेरै गाउँछन्. So then the clock is really fast. And here I'm plotting, like the dashed lines are 5 minutes and the colored lines are songs. So here you can see that the clock is really fast and also very regular at 9 minutes and 12 minutes. I want to pause here because this is remarkable, right? यसको मतलब के हो भन्दा हामीलाई अब हजुरहरूलाई कसैले ल मलाई 1 मिनट पास भएपछि भन भन्यो भने हामी अफ हुन्छौँ क्या. Like by a few seconds, like we don't even keep track of 2 minutes with this much accuracy. उनीहरूले चाहिँ 12 minutes, होइन 12 minutes को duration एकदमै accurately राखिरहेछ, right? So what this tells you is like in their brain दिमागमा एउटा सर्किट छ जसले चाहिँ टाइम 12 minutes को टाइम किप गर्न help गरिरहेछ, right? So this is very remarkable. Cool. So I wanted to quantify this, होइन यो अब क्लक जस्तो छ तर during the day मैले कसरी यो आवर चाहिँ क्लक लाइक छ यो आवर क्लक लाइक छैन भनेर कसरी assign गर्ने भनेर we came up with an index called the CLI, clock like index. अनि यसले चाहिँ के गर्छ भन्दा हजुरले dash line perfect clock हो, colored lines songs हो. So यो चाहिँ you can see एकदम alignment छैन होइन यो overlap भएको छैन. So the clock like index is very low. So in the same way, if I keep showing you different clock like index, your 0.96 is so close to 1. So it means like almost perfect clock likeness, एकदमै regular भनौँ न. यहाँ चाहिँ नि you can see there's really good alignment. अलिकति noise छ but it's really good, right? So this is what the formula of the clock like index is. So basically यो चाहिँ के भन्दा चाहिँ percent error जस्तो होइन हामीले error sum up गर्ने अनि त्यसलाई चाहिँ divided by the number of songs गर्ने जस्तो मात्र. It's a very simple metric. If you have questions, you can also reach out to me via email and I can tell you more about it. And so we developed an algorithm that allows us to study clock like index. Okay, and we looked at our data and we wanted to see कतिवटा songs are clock like, and this is a distribution. And what this tells us is like 0.8 अथवा 1 भनेको चाहिँ एकदमै clock like, 0 भनेको चाहिँ not clock like. So what we see, you can see that that 8 तिर 9 तिर it's very heavy, which means that it's very clock like. Okay, and here I'm plotting the songs. So each row is a day and each stick is a song. And the red color is a very high clock-like index, which means it's very clock-like, right? So you beat some that like it's very regular, right? So this is how I quantified clock-like index. Yeah. So that your solo setting, isolated setting, what we found is that this behavior spans multiple time scales. उनीहरूको diurnal, circadian and ultradian rhythm छ, which means that उनीहरू दिउँसो active छ हैन, अनि they sing at regular intervals, which we call clock likeness. So उनीहरू एकदमै like every 12 minutes or like every 10 minutes उनीहरू they're singing, right? Okay, so this is in the solo context. अब social context मा हेरौँ, त social context मा के हुन्छ? I told you पहिला like उनीहरू आफू पनि गाइरा हुन्छन् तर अर्को मुसा आयो भने पनि they interact with the other mouse, right? So like I said, just a reminder, your solo setting मा they can't hear each other. When I open the box, they can hear each other. So what happens when we open the box, right? So here I'm plotting 2 days of a solo interaction. एक्लै छ. Green is one mouse. Purple is another mouse, and you can see they have different rates, right? The green एकदमै fast गाइरहेछ, purple एकदम slow गाइरहेछ. So what happens when I open the box, right? एउटाले अर्कोलाई follow गर्छ कि दुवैजनाले आफ्नो rate change गरेर they come to a middle rate. What happens, right? So we see that only one mouse completely adjusts its rate to match the other. So your green ले चाहिँ केही change गरिरहेको छैन, but purple is completely changing its rate to match the leader. So here again, I'm plotting. In green is the end of the leader's song. Purple is the beginning of another song. And you can see your green ko song end huni bittikai purple le gaउँदो रहेछ. But if I flip it, मैले green को end मा purple को end मा हेर्यो भने चाहिँ green ले follow गरिरहेको छैन, which means that only one mouse is following the other. So एकजना चाहिँ leader, एकजना चाहिँ follower जस्तो role लिइरहेको छन् यिनीहरूले. अनि यो चाहिँ मैले probability of song plot गरेको. अनि what you can see is that the follower follows the leader but not the other way around. अनि मैले चाहिँ कति percent leader को कति percent song सँग follow गर्छ अर्को माउसले भनेर हेर्दा चाहिँ नि 67% भन्दा बढी songs in the first interaction is being followed by the follower but not the other way around. So and here I'm showing that the leader does not change its song rate in solo and social setting, but the follower changes its song rate in the social setting, not in the solo setting. All this to say that there's an asymmetry in social interaction. Again, here you see song length, song कति लामो हुन्छ भनेर हेर्दा followerले चाहिँ एकदमै compared to its solo setting, follower changes its rate a lot. Changes its song length a lot in the social context and not in the solo context. So there's an asymmetry there's an asymmetry in behavior, right? So एकजना चाहिँ leader, अर्को चाहिँ follower जस्तो role लिने रहेछ social context मा, right? अनि आफ्नो जुन solo rate छ त्यो solo rate को comparison मा उनीहरूले social मा एकदमै change गर्ने रहेछ, right? Okay. I'm going to leave out the normative theory for the interest of time, and I'm going to talk about the model. So okay, we have हाम्रो एकदमै cool behavior छ, right? So हामीले यसलाई mathematically कसरी describe गर्ने त, right? So we came up, right? So how do we capture this dynamic song patterning mathematically? So we came up with a statistical model. It's called a GLM-HMM. So the one, the generalized linear model, hidden Markov model. It's an intimidating name, so I'm going to go over what that means. I just want to tell you that यो मोडलले चाहिँ झिङ्गाहरू ड्रोसोफिलाहरूको courtship behavior study गरेको छ अनि miceमा decision making study गरेको छ. So it's a very flexible model that's been used in a variety of neuroscience studies. Okay. So let's look at our data again, right? Like I told you, your yellow चाहिँ नि daytime हो अनि यो white चाहिँ night हो. And you can see at night there aren't a lot of songs, right? एकदमै quiet छ, there's not a lot of songs. During the daytime, there's a lot of songs. And there's also a clock-like period. You can see in the morning it's very dense. In the afternoon, there's not a lot of songs. So यो चाहिँ कस्तो भन्दा चाहिँ states जस्तो लाग्यो क्या हामीलाई हैन. एउटा चाहिँ sleep state भनौँ न. Sleep state मा धेरै songs छैन. Clock state मा एकदम clock like songs छ. अनि maybe there are some states where एकदमै slow rate छ, अर्कोमा fast rate छ, right? So there are these states. How do we model these states, right? So हामीले चाहिँ यो GLM भनेको चाहिँ नि एउटा filter जस्तो सोचौँ. I can see my mouse. Okay, here it is. Filter जस्तो सोचौँ, right? So let's say हाम्रो एउटा weights को filter छ, हैन? यहाँ यस्तो अब पिक्स भनौँ न, एउटा रिपिटेड पिक्स छ, अनि जब हाम्रो songs past songसँग अलाइन गर्छ, we take what is called a linear combination. त्यो लिएपछि हामीले के गर्छौँ, probability आउँछ, अनि we roll a dice and we say is there a song or no song. यो perfect alignment भयो भने चाहिँ there will be a song. If there is not a good alignment, अलिकति यो filter र songs को यो perfectly overlap भएन भने चाहिँ probability is really low and there's no song, right? So यसमा चाहिँ there's a lot of things that happen under the hood, but this is how we're able to get clock-like songs. In the same way, we can get random songs. So your weights, like at the end, if we change the weights, the filter, we can make it such that it generates random songs. We're just rolling a dice and these songs randomly आइरहेछ. The same way in the sleep state, the weights are very low. Weights very low भएपछि चाहिँ नि हाम्रो songs नै हुँदैन, right? Okay. So now we have these states, right? Sleep state, random state, clock state. अनि यो मोडलले के गर्छ भन्दा चाहिँ it allows us to go from one state to another. So I can go from sleep to sleep, clock to random from sleep, random to random, and so on. So हामीले चाहिँ यो स्टेट्सहरु लूप गर्न मिल्यो. So that's what the model is. So at a given point, let's say म अहिले I'm in my talking state. So talking state पछि चाहिँ मा भइसकेपछि चाहिँ मेरो output चाहिँ vocal भयो हैन. So that's a vocal state. So एकछिन पछि maybe when you are going to ask a question, it's going to be a question state. And त्यो बेलामा maybe I'm going to be quiet and you're going to talk and answer questions. So we're just assuming these states and the mouse's output depends on these states. So यसरी चाहिँ हामी mathematically model गर्न मिल्छ. यो मोडलले मैले after I run it, after मैले parameter optimize गरेपछि चाहिँ I can generate data that looks like a real song. So मैले mathematically implement गरिसकेपछि this यो मैले देखाएको चाहिँ अघि नै मैले मुसाहरुको behavior देखाएको थियो. It looks very similar to that. And again, here is the clock, the minute scale, the zoom गरेर minute scale, we can also replicate real data. So all of this is data I generated from the model. In the same way, यो चाहिँ दुईटा माउसको interaction हो, यो माथि चाहिँ real हो, तल simulated मतलब मैले generate गरेको data हो, and you can see that I'm able to capture these dynamics using the model. Yeah, so that's aim 2. I think I'm good on time. I was able to capture these singing behavior using the GLM HMM. The third aim, which is really exciting that I'm doing, is like अब चाहिँ brain मा गएर, right? We have a lot of tools. Tools use गरेर we can disrupt brain regions, we can record from brain regions, अनि उनीहरुको यो गाइराको बेलामा मैले ब्रेन रेकर्ड गरेर कुन ब्रेनको कुन पार्टले यो बिहेभियरमा हेल्प गरिराछ हैन, यो सोशल इनपुट जुन छ नि त्यो अर्को मुसा आयो भनेर थाहा पाउने, those things using a lot of experimental tools. So that's what I'm doing currently in the lab. Yep, with this the summary is like यो singing mouse को उनीहरुले एक्लै हुँदा नि गाउँछन्, right? अनि त्यो एक्लै हुँदा गाउने चाहिँ it really depends on their internal state, the biology of their internal state. अनि त्यो चाहिँ धेरै एउटा टाइम स्केलमा पढ्न मिल्छ. The other thing is like उनीहरु एक्लै हुँदा र social condition हुँदा they have very different songs. अनि usually social interaction मा चाहिँ एकजना leader एकजना follower हुन्छ. अनि I'm able to use a mathematical model behavior. Okay, so with this, I would like to thank Cold Spring Harbor. First of all, I really want to thank Rajendra ji, who reached out to me when he was in Cold Spring Harbor and invited me to join NIRI and also give this webinar. So I'm really, really grateful to you. I'm also grateful to two of my mentors. This is Arka. He's an experimental neuroscientist of the background in biophysics. This is Ben Kali. He is a computational neuroscientist, and they've been really helping me with the project, with their mentorship. I'd like to thank the Banerjee Lab, especially Mike, Cliff, and Martin, who have been assisting me with my experiments, and everyone in the Kali Lab. I would also like to thank my family for their constant support, and I'm really happy to take questions.
Thank you so much, Yaman. Can you stop your
Yes.
screen so that we are all in the stop गरिदिनुहोस् त सेयरिङ चाहिँ भयो भने हामी सबै स्क्रिनमा आउँछौँ थ्याङ्क यू सो मच स्टप भएको छ अहिले होइन यही भएको छ होला ए हस् थ्याङ्क यू अनि केही क्वेसन छ भने प्लिज आस्क है
एउटा क्वेसन रहेछ च्याटमा Bilon asked, very interesting model to study behavior. Thank you for sharing. I'm wondering if there are any gender or age specific differences in the model. Oh, that's a really, really good question. That's a really good question. So someone in the lab is actually studying that. So from my experience, why does this question matter, right? Like, why is this a really key question? Is that in a lot of animals, हाम्रो चाहिँ के हुन्छ भन्दा चाहिँ मेल फिमेलको डिफरेन्सेस हुन्छ and that helps with reproduction, right? So in the singing mice, we see that even males, females both reply to each other. Females reply to each other, males reply to each other. So there's in terms of replying and following, there doesn't seem to be a difference, but males sing more than females. We've seen that. And the age specific difference is also a really good question. Someone in the lab, their whole PhD is related to that question. So हाम्रो के हुन्छ भन्दा developmentमा हाम्रो brainमा त्यो neural development is very different. So with age, what we see is that when they're very young, they don't sing full songs. They sing smaller songs, and as time goes, they sing longer songs. And one of the person in the lab is studying whether यो हाम्रो जुन leader follower वाला role छ, यो चाहिँ learned हो कि उनीहरूले अर्को मुसाबाट सिक्छ कि आफै नै is it just innate or is it learned भनेर. So that's a really good question. The answer is yes. There are gender and age specific differences, and someone in the lab is studying. So हाम्रो ल्याबको पेपर्सहरू हजुरले पढेर you can learn more about it. Great question.
Thank you, Yaman. Just to add, Dr. Bilan Khombu is an assistant professor at Tulane University. हाम्रो NIRI वेबिनारको फर्स्ट गेस्ट स्पिकर पनि हुनुहुन्थ्यो तारा सरले हात उठाउनुपर्छ त्योभन्दा पहिला चाहिँ निरजजीको क्वेसन छ त्यहाँ च्याटमा निरजजी हजुर चाहिँ इफ यु क्यान आस्क योरसेल्फ भिडियोमा आएर वुड बी इभन ग्रेटर
Thank you, Randy. I won't be able to turn my head. I'm still watching soccer. It was a great talk. I really appreciate for her, and it's a lot of information there. So this reminds me actually about there's one news about the Tony Bennett singing even after the Alzheimer, yes, because he picked the right notes and everything. So I was just wondering, like, how your model can be utilized on the translation research. Yes, ultimately we look for a translations. Yes,
right.
So I was wondering, like, because this is more on the neuro side, yes, right. And more on the neuro diseases. So just wondering. And by the way, thanks a lot. Really, good talk. Actually, I appreciate it.
Yeah, thank you so much. That's also a really good talk. So in order to do translational research, right, we have to first understand the circuit, right? Like we were able to cure a lot of diseases. We target proteins, right? The reason we're able to do that is because we know how the system works, right? Alzheimer's condition it's a very multidimensional problem, right? Like there's cellular deficiencies, and that changes the circuit, which in turn changes the behavior. So I think हामीले यो जुन circuit राम्ररी understand गरेर हैन हाम्रो motor interaction, speech कसरी facilitate हुन्छ, कसरी run हुन्छ भनेर, if we understand that, we can also understand the cell types involved in that circuit. And with cell types, we can understand, okay, what genes can we target? What proteins, if they're being expressed, can we target to understand this disease? So I really think what research like this, so in the singing mouse, right? Like it's also about evolution of a behavior, right? Like हाम्रो जुन मुसाहरू देख्छौँ हामीले जुन the mouse that we're familiar with, they don't have this novel behavior. So we can study this circuit and understand how a new behavior evolves and how the circuit changed. And once we understand the circuit, we can you can understand cell types, and we can utilize this in understanding how these circuits are involved in disease, right? And then we can target target and find targets for treatment. That would be my answer. Yeah.
Thank you.
Thanks a lot.
And Neeraj, just to add, I think it's very important to introduce as well. Neeraj Shrestha, Dr. Neeraj Shrestha is also a biologist. फ्लोरिडामा हुनुहुन्छ। हामी नजिकै छौँ। अनि उहाँ चाहिँ नेप्लिज एसोसिएसन अफ फ्लोरिडाको चेयरपर्सन पनि हुनुहुन्छ। एकदमै यो युथहरूलाई अनि फ्लोरिडाको स्टुडेन्ट्सहरूलाई कनेक्ट गर्ने, जोइन गर्ने। Thank you so much, Neeraj, for joining us.
Thanks, Hansi.
थ्याङ्क यू। अनि अब चाहिँ अर्को तारा सरले हात उठाउनु भएको छ, त्यसपछि हामी चार्टको क्वेसनमा जान्छौँ आशिषजीको। तारा सर, प्लिज टु यू।
हजुर, थ्याङ्क यू रायनजी, अनि थ्याङ्क यू एभ्रिवान। थ्याङ्क यू एमन फर अमेजिङ प्रेजेन्टेसन, एन्ड दिस इज सच अ न्यू topic for me. We get to hear about biochemical alterations, genetic alterations. Never heard about singing mice, let alone the research that's this circadian rhythm. So, मेरो क्वेसन चाहिँ निरजीले सोध्नु भएकै लाइनमा छ। So eventually, what has been done and what is being studied should be translated into human research. अनि माउस मोडलहरू भन्ने बित्तिकै हामीले इन्टरभेन्सनहरूको कुरा पनि सोध्छौँ होइन। माने आई वाज लाइक वाइल लिसनिङ टु योर प्रेजेन्टेसन, आई वाज द्याट क्वेसन केम टु माई माइन्ड, लाइक इफ यु वर टु इन्टरभेन the behavior or manipulate, then what would be those things, just out of curiosity?
Right. That's a really good question. That's a really, really good question. So that's like aim 3, right? So what we can start with is, this is really some of the ways we can do it is your muscles involved in like musakuma muscles involved in So there are tools, there are viruses that can allow you to track the circuit, like go back. So rabies virus can go back. You can trace the circuit of vocalization, right? And that way you can understand what circuits are involved. So already मेरो PI ले चाहिँ उहाँको old postdoc workमा चाहिँ I think someone else. So उहाँको postdoc researchमा they studied that there are brain regions that are involved in singing. So what he did is he perturbed उहाँ चाहिँ he lesioned the brain region भनौँ न। अनि त्यो गरिसकेपछि मुसाले चाहिँ एकअर्कासँग रिप्लाई गर्न नसक्ने हैन। Someone in the lab is also studying this brain region called the periaqueductal gray area. त्यो गरेपछि पनि उनीहरूले vocalize गर्न सक्दैनन्. So we actually know these circuits that are involved in vocalization. अनि त्यो activate inactivate गरेर चाहिँ we can actually, a really tough question to answer in my project is to 12 minutes सम्म उनीहरूले टाइम कसरी keep गरिरहेछ. That's a very tough question to answer, right? Because neurons, they operate in milliseconds, right? So how can you go from milliseconds to minutes, right? That's a really interesting question, because in circadian rhythm, you have this transcription factor feedback loop, like a technical bio, but like biochemical reactions and so, which are at 24-hour scale. They've evolved to be at 24-hour scale, but minute scale ko isto त्यो behavior so we don't know of. But there are candidates, and like I said, हामीले त्यो neural tracing गरेर we can trace the circuit involved in vocalization, and then we can go in अनि हामीले त्यहाँ manipulate गरेर perturb गरेर अथवा activate गरेर over inactivate activate गरेर we can see how that changes the behavior, and then find a causal relationship with the biology and the behavior.
Thank you so much. Beautiful answer. And you are only on your 3rd year of your graduate research. I can see how it could expand and explode as time passes by and people like you are involved. My best wishes, Yaman, and thank you one more time to come and present to us.
Thank
Thank
you.
you.
थ्याङ्क यू सर अनि एउटा सञ्जयजीले फेसबुक लाइभ च्याटमा पनि यस्तो ब्युटिफुल क्वेसन सोध्नु भएको छ सञ्जयजी अल्सो देयर तर सञ्जयजी म हजुरसँग एकछिनमा आउँछु लेट्स टेक वान क्वेसन फ्रम द च्याट पहिला थियो त्यहाँ आशिषजी हजुर चाहिँ इफ यु आर हियर आई थिङ्क यु आर हियर कुड यु आस्क द क्वेसन यर सेल्फ हजुरले नै सोधिदिनु भयो भने राम्रो हुन्थ्यो
Hi, so amazing presentation. Thank you. That was really, really beautiful. You are studying circadian rhythms and they are connected to dopamine, I believe. So any thoughts on dopamine, its involvement in its behavior, and if we can, then how can we apply this?
That's an interesting question. So dopamine is involved in a lot of different things. So when we talk about circadian, so there's two things, right? There's causality and there's correlation, right? So there are certain things that will cause, like, you know, if I just drink coffee, I'm going to be agitated, right? So that's causal. Some things are correlational, right? So there might be things that dopamine is a neurotransmitter. So there might be things that as you go throughout the day might be going up and down. Whether that is causal or correlated, we need more things to determine that. So I think with circadian rhythms, there are things like melatonin and other neurotransmitter that are causal. So dopamine, circadian bhanda pani, there's ultradian rhythm ma चाहिँ it is known to be correlated. So ultradian भनेको चाहिँ several hours वालामा, right? So there we know there's a dopamine oscillator that goes up and down at several hours. So that I am actually really curious in studying in studying its involvement in the behavior. So what I mean by this is like what we see in lab mice is when their dopamine changes throughout the day, unarko activity, like how much they move, is also changing, right? So I think in the same way I've looked at behavior, like singing behavior and locomotion, and I can see that they're very correlated. So to answer your question, at the ultradian scale, not necessarily the circadian scale, I don't know about that, but like the ultradian scale, the dopamine has been shown to be, dopamine might be involved in the activity and arousal state that affects the number of songs that they sing. And how can we apply this to the behavior, right? So I really think that like in Parkinson's, right, there's dopamine is affected, and that affects your movement. So if we can study in the involvement of dopamine in movement at minute scale, which I want to do, then we could really maybe perhaps utilize this in informing treatments in diseases such as Parkinson's.
Thank you.
Thank you. Our next question, एकदमै interesting question छ सञ्जयजी. Sanjay ji also is with us at NIRI. Thank you, Sanjay ji. Please ask the question. हजुरले नै सोध्नु त.
Thank you, Rajendra ji. Yaman bhai, I really appreciate the presentation you made. पहिला कुरो मलाई चाहिँ अब यस्तो प्रेजेन्टेसन के मेरो सब्जेक्ट म्याटर हो नि क्युरियस हुन मलाई हेल्प गर्छ. द्याट्स व्हाट आई लाइक अबाउट दिस. यु हेल्प मी टु बिकम क्युरियस. मेरो एउटा क्युरियस क्वेसन के छ भने जस्तै माइसहरुमा त नट अल माइस आर सिंगिङ इक्वली वेल. प्रोबब्ली त्यो स्ट्यान्डर्ड डेभिएसन मिनहरु हेर्दाखेरि हामी ह्युमन्समा पनि के हुन्छ सम पिपल आर डुइङ समथिङ स्किल्स बेटर एन्ड वी पिक द्याट पर्सन टु बी द लिडर, राइट? माइसहरुमा पनि त्यस्तो हायरार्की हुन्छ ताकि उनीहरुले सेल्फ रिअर्गनाइज गर्छ क्या उनीहरुले, ताकि ए माइस वान इज सिंगिङ बेटर ओभर टाइम सो लेट्स चुज द्याट माइस वान एज अ लिडर भन्ने त्यस्तो लिडरशिप हायरार्की देखिन्छ माइसहरुमा.
That's a really good question. That's a really good question. मैले एउटा कन्फरेन्समा प्रेजेन्ट गर्दा नि everyone was like, oh my god, leader follower in songs, how does that affect hierarchy, right? So the really cool thing is that the short answer is I don't know. The longer answer is that उनीहरूको यो vocalization छ नि यो long distance हो क्या. So when they're closer together, they don't really sing like this. They have another mode of vocalization जुन चाहिँ अरू हाम्रो जुन हामीलाई थाहा भएको मुसाहरू छ नि उनीहरूसँग चाहिँ ultrasonic vocalization छ. यो मुसाहरू पनि ultrasonic vocalization पनि बनाउँछ. When they're closer together, they switch to the second mode. When they're at a longer distance, they sing. So उनीहरू चाहिँ एउटा Costa Rican, like imagine a really dense cloud forest. So maybe your smell is impaired, your vision is impaired, so that they develop this long distance thing. So maybe it's not involved in hierarchy, but rather like calling out the other person. But having said that, we haven't tested hierarchy, right? So people who study hierarchy, what they do is they take a tube, they take two mouths, अनि उनीहरूले like ट्युबमा छोडिदिन्छ. लिडरले चाहिँ एउटा माउसलाई ढकेलेर अर्कोबाट निकालिदिन्छ क्या. So that's how you determine leader follower. So in our case, the leader follower is very consistent. I've looked at variables such as like हजुरको song length, हैन, हजुरको song कतिवटा song गाउँदो रहेछ भनेर. अनि initially I was like, oh, जुन मुसाले चाहिँ कम गीत गाउँछ, like कम number of songs गाउँछ, उ चाहिँ usually leader हुने रहेछ भनेर observe गरेको थिएँ. But then I found another pair where that was not true. So, you know, that's a really interesting question, but the short answer is it isn't obvious as to which variable it is contributing to that behavior. But what we could do is we could use machine learning, and what we can study is like हामीले नबुझेको नि हुन सक्छ नि त, right? Like त्यो information song को structure हामीले त length हेर्छौं, कतिचोटि गाउँछ हेर्छौं, loudness हेर्छौं. But if there's a higher dimensional structure in the song, then we can't infer that with just our human intuition. So we can use machine learning models. So someone in the lab actually used a model called variational autoencoder. उनीहरूले त्यसले चाहिँ के गर्छ भन्दा songलाई decompose गरेर त्यसको latents भनौं न hidden variables देखाइदिन्छ. त्यो देखेर चाहिँ के थाहा भयो भन्दा चाहिँ हजुरको different mouse को songsहरू different clusters मा आउने रहेछ क्या. So what that means is songs have information that can maybe signal something like hierarchy, but maybe it's not that intuitive to humans. But that's a great question. Yeah, thank you.
एउटा चाहिँ क्विक सन्देशजीकै क्वेसनमा एउटा पोइन्ट एड गर्छु अनि म आई कम टु सुभाषजी. उहाँले हात रेज गर्नुभएको छ. यो नेपालको नेताहरू जस्तै हुँदो रहेछ, कसलाई धक्केलेर बाहिर निकाल्ने, कोही कम बोल्ने, कोही धेरै बोल्ने भने जस्तो के. सो मच हामीले यस्तो रिलेटेड गर्न सक्दो रहेछौँ कि लिडर चाहिँ कस्तो हुन्छ भन्ने कुरा चाहिँ. थ्याङ्क यू सञ्जयजी, सच एन अमेजिङ क्वेसन. अब सुभाषजी हजुरले हात उठाउनु भएको छ. सुभाषजी इज अल्सो NIRI मेम्बर. प्लिज गो अहेड.
थ्याङ्क यू राजनजी. यमनजी भेरी इन्ट्रेस्टिङ टक. म मेटेरियल साइन्सको रिसर्चरलाई चाहिँ यो एकदम नयाँ हुँदो रहेछ. अनि मलाई एउटा क्युरियस लागेको कुरा चाहिँ जस्तै अब तपाईँले यो माइसमा स्टडी गर्नुहुन्छ होइन, यो तपाईँको आफ्नो प्याटर्नबाट. जस्तै हामीले अब यो माइसहरू त अब इभन मेटेरियल साइन्समा पनि हामीले ड्रग डेलिभरीहरूमा चाहिँ माइसकै मोडल युज गर्छौँ. अनि अब बायोटेक्नोलोजी यो अरू हेल्थ स्टडीहरूमा मेडिसिनको स्टडीहरूमा पनि माइसलाई युज गरिन्छ. त्यो माइसलाई जुन माइस हामीले युज गर्छौँ त्यसलाई अब त्यसको पनि त प्रकृति अब अहिले तपाईँको यो स्टडीबाट हेर्दाखेरि चाहिँ माइसको पनि प्रकृतिहरू हुँदो रहेछ विभिन्न खालको होइन. त्यो चुज गर्दाखेरि पनि यो तपाईँको प्याटर्नहरू स्टडी गरेर गरिन्छ कि ऱ्यान्डमली गरिन्छ होइन. अब अथवा मानौँ न हामीले यो तपाईँको स्टडीबाट माइसको क्याटेगोरी छुट्याएर त्यसलाई चाहिँ अलिकति अब एक्टिभ माइसलाई युज गर्दाखेरि हाम्रो रिजल्टहरू राम्रो आउन सक्छ होइन. त्यस्तो खालको स्टडीहरू पनि भइरहेछ कि के छ त्यसको बारेमा अलिकति तपाईँले जाने बुझेको कुराहरू सेयर गरिदिनु होला. हस्, थ्याङ्क यू. I think you're muted.
You muted, sir.
Sorry, thank you. So that's also a really, really good question. So मैले study गरेको mouse, even in the earlier plots I showed you, you can see that each mouse is very different. It's different in a stable way, right? So all of this is the brain, right? Like all of this variation in behavior is because of the brain. So what we can do actually is use, not like select the best ones, but use this variation to study the neural substrates or biological substrates, right? So there's a thing called quantitative trait loci. So it's a genetic technique, and what they do is like धेरैवटा traits छ, let's say you have different tomatoes, one tomato plants, one has 10 tomatoes, one only has 2, right? You can look at this variation, you can map what biological substrates led to this variation, right? So I think besides choosing the best ones, well, I tend to do that for my research because it's easier if I have a mouse that's singing a lot. But if I don't, like if there's variation that can actually inform us about differences in circuits, right? So मैले एउटा एकदमै एक्टिभ माउस र अर्को एकदमै इनएक्टिभ माउसको होल ब्रेनमा like activity imaging गरेर in one of the mouse there are certain regions that are very active versus not the others, so I can correlate this variation in behavior and in circuits to actually study them. So yeah, I think variation actually informs us about the biological substrates. That answered your question.
Yes, thank you. अरू केही क्वेसनहरु छ भने प्लिज आस्क, हामीसँग केही समय छ। मेरो एउटा क्वेसन छ, त्यो जस्तै स्याड हुँदाखेरि गाउने प्याटर्न्स र अनि त्यो ह्याप्पी हुँदाखेरि गाउने प्याटर्न्सहरु छ, अनि कहिले चाहिँ उनीहरुले फेरि स्याड हुँदो रहेछ, त्यो चाहिँ खास त्यो केसले साइन्टिफिकल्ली हेर्दाखेरि एक्सपेरिमेन्टली हेर्दाखेरि चाहिँ केले ट्रिगर गर्दो रहेछ त्यो स्याडनेसमा, किन त्यो हर्मोन्सहरु ह्युमनसँग मिल्छ कि त्यो त्यो केले ट्रिगर गर्छ उनीहरुको त्यो स्याडनेस चाहिँ, अनि त्यो साउन्डको डिफरेन्सेस के छ त्यो स्याडनेस र ह्याप्पीनेसको, अनि अरू त्यो स्याडनेसलाई चाहिँ अर्को माइसले कसरी रेस्पोन्ड गर्दो रहेछ ह्याप्पीनेससँग, त्यस्तो केही अध्ययन भएको छ हजुरको अध्ययनमा?
मेरोमा चाहिँ भएको छैन. I can tell you about other research, like really cool research that has been done in other mice. So उनीहरु चाहिँ if उनीहरु डराइरहेछ भने they don't sing at all, right? So but if they have plenty of resources, उनीहरुलाई चाहिँ insectivorous हो, उनीहरुलाई किराहरु दियो भने चाहिँ they sing a lot. So there's an underlying arousal state. So Ashish mentioned earlier the dopamine involved हो कि भनेर, like so if when you are in a aroused state, maybe there are neuromodulators, hormones that change, and त्यसले गर्दा बडी गाउने जस्तो हुन सक्छ. If they're scared, they don't sing a lot. So we can do things like that, right? Like we can induce some kind of fear or sadness भन्दा नि fear अथवा कुनै different contextमा we can see how their behavior changes. That's a really good question. I just want to say not in this species, but another species, I'm forgetting if it's voles or mice, they studied empathy. So what they did is they have 2 mice co-housed, अनि एउटालाई चाहिँ mouse or vole, whatever, a rodent, अनि they take one of the rodents away for 15 minutes and they shock the rodents, bring it back, and what you can see is the other mouse, the mouse that is the observer, that is not involved, that wasn't shocked, even their cortisol level and their neural activity is changed because of the interaction of the shocked mouse. So in a way, it's like they call it empathy, in a way that like you're relating to the other mouse. And there are actually, they impaired this thing called oxytocin receptor, which is in like a protein in the brain. त्यो impair गरेपछि they're not able to do this behavior. So you can actually study proteins and other genes and neuromodulators, or these molecules that are actually involved in this behavior. So singing mouse ma चाहिँ छैन, but a good way, a good experiment would be, we can take the mouse, we can induce different conditions, right? Like shock it or give it a lot of mealworms, social interaction, different setting, we can study and infer what chemicals are involved in this conditions, right? Yeah.
Thank you. Just wanted to take one quick opportunity to ask you one more question. यो चाहिँ अलि ल्याब बेस्ड जस्तो, ल्याबमा कति how easy or how hard is to handle those mice? जस्तै कोही माइसले एकदमै गाह्रो हुने टोक्ने गर्छ नि, मैले माइसमा काम गर्दाखेरि अनुभव गरेको।
एउटाले चाहिँ टोकेको छ,
हजुर भन्नुस् त। सरी,
एउटाले टोकेको छ हजुर। त्यो ल्याब माउस चाहिँ के हुन्छ भन्दा चाहिँ we have been breeding it in the lab for so long, so they're very friendly to handle. सिङ्गिङ माउस चाहिँ innate हो, अनि उनीहरू वाइल्ड नै छ अहिलेसम्म, there's only three singing mouse labs in the world. So they're very hard to handle in that sense. But we have tricks of, so we use a tube, cage tube. They love the tube, they go around the tube. So what we do is when they're inside the tube, we just close the tube and then we transfer them. So they're easy to handle in that context, but compared to lab mouse, not that much. And हाम्रो त्यति धेरै genetic tools पनि छैन for singing mice, but we can figure things out and use existing tools to do it. Yeah.
Yes, thank you. And अरू कसैले हात उठाउनु भएको छैन? That's our last question म सुन्न चाहन्छु। यो माउसहरू कहाँबाट लिएर आउनुहुन्छ हजुरहरूले चाहिँ, जस्तै bring कहाँबाट?
So चाहिँ Costa Ricaबाट हो. I think so NYU मा मेरो PI NYU मा काम गर्नुहुन्थ्यो, उहाँको चाहिँ पुरानो ल्याबमा I think they had brought some, I think, from Costa Rica, but after that we've been maintaining it in our lab, but they're native to Central America.
Yeah, good. Thank you. Tara sir, to you. हजुर म्युटेड हुनुहुन्छ।
यो एक्सपेरिमेन्टल कन्डिसन, त्यो इन्भाइरोमेन्ट जुन अरू रेगुलर माइसहरू राखेर रिसर्च गर्ने एनिमल फेसिलिटीहरू हुन्छन्, त्यो नै सफिसियन्ट छ कि अथवा यसको लागि विशेष साउन्ड प्रुफ, किनभने अब हजुरहरूलाई त्यो साउन्डहरू रेकर्ड गर्नुपर्ने हुन्छ होइन, ब्याकग्राउन्ड नोइजले इम्प्याक्ट गर्न सक्छ, त्यो एउटा कुरा। अनि अर्को कुरा फियरको कुरा गर्नुभयो, हजुरको रिसर्चमा त त्यो इन्भल्भ भएको छैन, तर रियली कुल आइडिया that how we can create fear or anxiety or that shock effect and study empathy. That's really, really cool idea. के छ भन्नाले हजुरको यो सेपरेट त्यो एनिमल फेसिलिटीमा पनि सेपरेट रुमहरू हुन्छन् कि एउटै फेसिलिटीमा गर्नुहुन्छ?
उसको सेपरेट रुम हुन्छ अनि तर सबै सिङ्गिङ माउस त्यही हुन्छ। हामीले रेकर्ड गर्दा चाहिँ जब हामी उनीहरूलाई कलोनीबाट बाहिर निकालेर हामी रेकर्डिङकोमा चाहिँ हामी सबै साउन्ड प्रुफ बक्स युज गर्छौँ। अनि अर्को के समस्या हुन्छ भन्दा चाहिँ उनीहरू एउटै रुममा छ भने, मैले एउटा एक्सपेरिमेन्ट गरिरहेको छु, मेरो साथीले अर्को गरिरहेको छ भने साउन्ड प्रुफ भएन भने उनीहरू एकअर्कासँग इन्टरेक्ट गर्छ नि त, अनि त्यसपछि भेरिएबल बिग्रियो। सो मोस्टली साउन्ड प्रुफ युज गर्छौँ। कलोनी चाहिँ इट्स सेपरेट फ्रम ल्याब माउस। ल्याब माउसको लागि छुट्टै एउटा रुम छ, singing mouse clay छुट्टै छ। Yeah, it would be really cool to study anxiety and other yeah. In mouse model, the thing with that is really so. The reason the way we classify these mental conditions is through service in human beings, right? Like we ask them questions. Questionnaire, we ask them, okay, how are you feeling about that? Then there are things you can do, like so one of the things is helplessness, right? So Musa, if you, the ones that are depressed, if you just tail suspension, if you leave them in water, they won't fight as much, they won't try to they'll just give up, right? That's one aspect. The other is like there are changes, cellular genetic changes that can lead to this, right? So it's studying depression and anxiety is really hard because how are you going to assess that they are depressed or they have this condition? What genetic perturbations are you going to use? So it's a really tricky thing, but it would be cool to see your different conditions, but how do their behaviors change, right? Yeah, that's a great idea. Thank you.
Yeah, thank you, Yaman. And so that's one thought. So it would be really nice to apply what is already known from the regular mice to this model, and vice versa. Vice versa. Yeah, it's really, really, really nice, cool. And I can already imagine how exciting it is for you to do this. Thank you.
So,
thank you.
Yeah, that just reminded me of something I wanted to tell you. So someone in the lab has actually done that, is compared lab mouse co-brain and singing mouse co-brain, right? And you can think we know of circuits that changed that led to one behavior and another. So that's something we're doing at circuit level. That's a great idea. Yeah.
Thank you, Emma. Yeah.
Thank you, Ajay.
Thank you, Andy.
Thank you. Neerajile haat uthanu bhako. Thank you, sir. Neerajile haat uthanu bhako cha.
I just want to, Azra, I just want a quick question actually. So your autism ko your case ma, have you thought of Have you guys thought of any publication or anything? Your autism because the autism concept. There's so many things happen, yes, and it's noise is one of the factor for these autistic people, yes. So I'm just wondering, like, is there anything you can add on this one? Probably connect with your research or somebody's doing in this.
Yeah, so I think I'm not super well versed in autism. I mean, it's not been studied in the singing mice for sure, but I know that people have done sequencing. So there's this thing called sequencing that tells you about all the genes that are active in a way, right? So people have done sequencing, compared mice with autism and not, and I think there are genetic changes, mutations you can introduce to induce autism, but I'm not super sure. One of the things that happens in autism is impairment of social interactions, right? So I think singing mice would be a great model to study that too. I'm not very well versed, but I think there are various studies ranging from behavior to studying sequencing or genetics, but I'm not super well versed in that. Yeah.
All right.
Sorry.
Thank
Thanks.
you. Thank you, Neeraj. Thank you, everyone. There is a कसै क्वेसन छ भने we can take 1 or 2 last questions. छैन भने चाहिँ I think हामीले लाइभ बन्द गरेर 1-2 minutes यतिकै केही इन्फर्मल्ली कुरा गरौँ अनि I think we'll be done then. Suraj, I think we can go out from live for now.